In the Star Wars movie Attack of the Clones, Padme and Anakin end up fighting Genosians on an automated assembly line. Padme manages to survive because the assembly line is set up in an ordered and predictable way. She knows when to jump, when to pause, and so on to survive. If there were more chaos in the line, she might have been killed. And then there'd be no Luke or Leia. Just like the assembly line on Geonosis, genes are set up in an ordered and predictable way. Nucleosomes, which are barrel-shaped clusters of proteins, are spaced along the gene's DNA to help keep it from getting tangled up. But when an enzyme called RNA polymerase II comes sliding along the gene, the nucleosomes need to be carefully removed in front and then replaced after it passes. Similar to the plight of poor Padme getting hurt if the assembly line were not predictable, bad things can happen if nucleosomes aren't taken on and off the gene in a timely manner. For example, if nucleosomes aren't removed, then RNA polymerase can stall. On the other hand, if nucleosomes aren't put back after being taken off, then the gene might mutate and become longer. In a new study in genetics, Koch and coworkers showed that the budding yeast gene ISW1 is involved in getting nucleosomes back on DNA after being taken off. When yeast lack ISW1, nucleosomes don't always return after being removed to make way for Pol2, and for some genes, this spells trouble. The genes that have the most problems are those that have something called triplet repeats, where 3 of the same bases, such as CAG, are repeated many times in a row. Koch and coworkers showed that a gene with triplet repeats ended up with more of them if ISW1 wasn't there to shepherd the nucleosomes properly. And this can be bad news. Too many triplet repeats can damage the gene and ultimately the cell. So why do the repeats expand? Koch and coworkers postulate that when cells are missing ISW1, nucleosomes aren't replaced as efficiently, and stretches of DNA can be left bare. If one of these stretches contains CAG repeats, it can end up forming new structures called hairpins. Cells like to send in DNA repair machinery to deal with hairpins, but unfortunately, the repair enzymes have a lot of trouble dealing with this kind of DNA. Like the Three Stooges, who tried to fix a plumbing leak by adding more pipes, repair enzymes can actually end up adding more DNA to deal with the hairpin. The results are not as hilarious as they were for Mo, Larry, and Curly. Too many triplet repeats can be harmful, not only to yeast, but to us as well. In fact, triplet repeat expansion disorders in humans happens when the number of repeats increases from one generation to the next. These devastating diseases mostly cause slow and steady degeneration of nerve and brain cells. The most well-known is probably Huntington's disease, which killed folk singer Woody Guthrie. Humans have similar proteins to yeast ISW1, so understanding how ISW1 prevents trouble with triplets in yeast may help us figure out how triplet repeat expansion happens in humans. If it turns out the same is true in people, then once again, yeast will have shown us how human genetic diseases might happen.